Good afternoon, everyone. Thanks to all of you for joining us for today's endocrine rundowns. Today, we are very pleased to have Dr. Zhenmin Lei talk to us about the functional role of androgens in regulation of inguinoscrotal testis descent. Dr. Lei is a MD PhD who obtained his MD degree from Fujian Medical College in China. He came to the United States, to Louisville, I think in around 1987 or so, and completed his PhD. He has been on faculty at the University of Louisville since '95. He's currently professor and vice chair of research in the Department of Obstetrics and Gynecology. He has extensive research record. He has trained over 19 postdoc fellows, has had 28 clinical fellows work with him on various different projects. He has had funding for a long time from the NIH, has over 150 peer-reviewed publications, particularly related to reproductive endocrinology, the role of LH and HCG. So today he'll be talking to us about androgens and their role in inguinoscrotal testicular descent. Dr. Lei.
Thank you. Okay, I do not have any financial or non-financial relationship to disclose. The cryptorchism, which in Greek means hidden gonad, the failure of the testicular descent into the scrotum, is a very common birth defect in males. The prevalence is about 3 to 5% in full-term newborns. It is estimated to be as high as 30% in the premature boys. Cryptorchism is a major risk factor for male infertility and for testicular malignancy. About 40% cryptorchid patients develop oligospermia to azoospermia. The incidence of the testicular cancer is estimated to be 4 to 7-fold higher than normal men. Although the etiology of the cryptorchism appear to be multifactorial, endocrine disorders are believed to play a major role. There are evidences to support that increased prevalence of cryptorchism among boys with the conditions associated with the congenitally decreased secretion or action of the androgens. The diseases or syndromes associated with decreased androgen level or androgen action may include the following 4 categories. First, disorder of sex development. The sex chromosome disorders such as 47XXY, 46XX males, steroidogenic defect. This included a number of the steroidogenic enzymes gene mutation or steroidogenic factors such as STAR gene mutations, androgen receptor mutations, LH receptor defect. Second, the congenital hypogonadotropic hypogonadism in 46XY, such as common syndrome. That include more than 20 genes mutations such as FGF1, FGF8, POC1, POCR1, etc. And pituitary hormone deficiency. Third, congenital hypergonadotropic hypogonadism, such as Down syndrome, as an extra copy of chromosome 21. Fourth, syndromes associated with both primary and secondary hypogonadism, such as Prader-Willi syndromes. Although androgenic action is believed to be essential for normal testicular descent, the cellular targets of the androgen action, the local cell signaling pathway, the proliferation or differentiation stimuli activated by androgen are not fully understood. This slide illustrates the cause and the hormonal regulation of the testicular descent. The testicular descent can be anatomically divided into two phases. Namely, is the transdermal phases, the first phase, and Inguinoscrotal, or they also call transinguinal phases. That's the second phase. Although the timing is different between humans and rodents, but the process is similar. In the human, the testicular descent is complete before born. In mouse, the testicular descent is not complete until postnatal 14 to 21 days. Initially, developing testes is located near the cranium, anchored by a cranial suspensory ligament connected to the diaphragm. On the top, the bottom is another ligament called cubiculum. The testosterone secreted by testes act on cranial suspensory ligament to accelerate this ligament to migrate. Meanwhile, the insulin-like growth, insulin-like factor 3 activate the ISFP2 signaling in the cubiculum to stimulate the cubiculum to growth and enlarge. As such, the testes is moved from abdominal location to the future inguinal canal area. This is the first phase of testicular descent. During the second phase of the testicular descent, the cubiculum continues to grow and enlarge. The outer rim of the cubicular cells start to differentiate into the muscle cell and grows inversely upward to form a sac structure called cremaster muscle sac that allows testes to descend. The expansion and enlargement of the cremaster muscle sac causes the dilation of the inguinal canal that facilitates the testes to extend to the scrotum. With respect to hormone regulation, the current consensus is that androgen, the testicular descent during the second phase of testicular descent, is androgen dependent. However, the mechanism by which the androgen regulates the second phase of testicular descent is still elusive. Where the testosterone acts on cubiculum to regulate cubiculum development and differentiation or elsewhere is still controversial. Moreover, whether or not the insulin-like ISFP2 signaling will play a role during the second phase of testicular descent remain to be clarified.
Stand in front of the camera so people can see you.
Okay. Okay. So a few years ago, our lab generated the LHCG receptor knockout animal. The LHCG receptor inactivation handles the latex cell development, resulting in the androgen deficiency. So the panel A shows that the latex cell that immunofluorescence staining shown in the pink color, located in the interstitial cell of the testes in wild type animal. The LHCG receptor knockout resulted in the absence of the latex cells in the testes. Therefore, the serum testosterone level in the LHCG receptor knockout animal is extremely low, barely detectable by radioimmunoassay. This slide shows the gross appearance of the genitalia of the adult LHCG receptor knockout male mice. The picture displays that both sides of the testes are descended into the scrotum. This is scrotum here, this is penis. In the wild type and the heterozygous animal, where LHCG receptor knockout mice, both testes are trapped in abdomen. The panel A of the picture shows the well-developed cremaster muscle sac holding the testes in the scrotum in wild type and heterozygous animal. The underdeveloped cremaster muscle sac in the LHCG receptor knockout mice is too small to accommodate the testes. Even though the size of the testes in the LHCG receptor knockout is much smaller than that of the wild type. Histological examination revealed the development of the gubernaculum in the postnatal day 1 to postnatal day 7 animal is similar between the wild type and the LHCG receptor knockout. The development defect of the gubernaculum becomes obvious from the postnatal day 14. As you can see, the picture here shows the gubernaculum bar contain the undifferentiated basophilic mesenchymal cells in the LHCG receptor knockout, and this is persistent into the adulthood. On the other hand, in the wild type animal, in this stage, most of those mesenchymal cells has been differentiated into the muscle cells. The 21 days of testosterone replacement therapy stimulate gubernaculum to grow and differentiate it, and normalize the size of the cremaster muscle sac, and induce the testes descent into the scrotum, as shown in here. This is LHCG receptor knockout animal after the 21 day of the testosterone replacement therapy. This well-developed cremaster muscle sac holding the testes in the scrotum. The same as the classic androgen target tissue epidermis, the RT-PCR and the Western blot demonstrate that the gubernaculum also expresses androgen receptor genes. The immunohistochemistry staining shows that the androgen receptor protein is localized in the nuclei of the gubernacular mesenchymal cells as well as the cremaster muscle cells, suggests that the gubernaculum is the target of the androgen. The Anguinic laboratory recently reported that selective inactivation of the androgen receptor gene in the gubernacular mesenchymal cells resulted in the Inguinoscrotal testicular male descent. This is the same phenotype as we observed in the LHCG receptor knockout. So this study provides further evidence to support that the directly action of the androgen on the gubernaculum is essential for the second phase of testicular descent. So this slide recap the results so far. The luteinizing hormone receptor knockout resulted in androgen deficiency, and male mice exhibit bilateral inguinoscrotal quadric phenotype. The testosterone replacement therapy completely restored the inguinoscrotal testis descent in LHCG receptor knockout males. The gubernaculum is a direct target of androgen actions. To investigate the underlying mechanism by which androgen in the second phase of testicular descent, we first set out to profile the global androgen responsive gene. The 14 years old of the LHCG receptor animal was received a single testosterone injection. 24 hours later, the RNA is isolated from cubiculum tissue and hybridized to the affirmatic the mouse genome array chip. The microarray analysis shows that the LHCG receptor knockout resulted in the upregulation of 19 genes and downregulation of 8 genes. The testosterone replacement therapy upregulated 46 genes and downregulated 27 genes. And this is a heat map of some of the differentially expressed genes not regulated by androgen injections. As they show in here, for example, IXFP2, in the knockout animal, is significantly decreased as compared to wild type. Then after the androgen administration, this expression of these genes in my was significantly upregulated. Another one is APP1 on the other hand, the mRNA level is significantly higher in the knockout animal compared to the wild type. After the testosterone treatment, is downregulated. This table lists the 8 of most affected the genes either downregulated or upregulated by testosterone treatment. And the microarray data is further confirmed by real-time PCR. And here it shows there's 2 of significant downregulated genes in the knockout animal, XMOC1 and IXAP2, and the 6 of upregulated genes in the knockout animal. And those genes expression can completely reverse by testosterone treatment. Very interestingly, in those genes, only IXFP2 is the one known to be critical for the first phase of the testicular descent. Whether or not the IXFP2 still play a role during the second phase of testicular descent remains to be determined. So what's the IXFP2? The full name of IXP2 is relaxin family peptide receptor 2. They're also known as LGI8, or another name is GREAT, GREAT. It's a leucine-rich repeat containing G protein couple receptor. The ligands of IXFP2 are insulin-like peptide 3 and relaxin. Relaxin and the insulin-like peptide 3 are protein hormone with 2-chain structure similar to that of insulin. IXFP2 mediate the action of the relaxin or insulin-like peptide 3 through a cyclo-AMP dependent pathway. The insulin-like 3 relaxin IXFP2 signaling is necessary for the first phase of testicular descent. The mutation of the IXFP2 gene in human or deletion of IXFP2 gene in mice all result in first phase of testicular descent failure. To better understand the role of the IXP2 during the second phase of testicular descent, we first determine the temporal and spatial expression of IXP2 protein in the cubiculum. The result shows that the IXP2 expressed in the cubiculum mesenchymal cells as well as in the cremaster muscle cells. The IXFP2 protein expression is gradually increased with age. As I show it here, see, this is wild type A1, A7, A14, this adult shows with age increase of the IXFP2 protein level. This increase is significantly impaired in the LHCG receptor knockout. As you can see that they even come to the A14 or adult, but adult the LHCG receptor knockout, the IXFP2 protein level in the cubiculum or cremaster muscle is very low. The 21 day of the testosterone replacement therapy completely reversed the IXP2 protein level to comparable to that of the wild type. To determine whether or not the IXFP2 expression is directly regulated by androgens. So we perform an in vitro organ culture, treated those the molecular tissue with dihydrotestosterone (DHT), and the result shows that the dose and the time-dependent upregulation of the IXP2 expression by DHT in culture HCG receptor knockout molecular tissues. Moreover, co-treated and the androgen receptor antagonist flutamine also block the DHT induced the IXP2 expression, so suggests that this is androgen receptor mediated actions. The short-term in vivo androgen, the testosterone injection also enhance the molecular cell proliferation, as showing that the increase of the BRDU incorporation. Those brown that is represents the nucleus of the cell is undergoing DNA replication. So as you can see, the testosterone injection increases cell molecular cell proliferation. Very interestingly, if we inject the IXP2 inhibitor, IXP2 inhibitor is a synthetic insulin peptide 3 analog. It competes with the insulin peptide 3 for binding to the receptor IXP2, but cannot activate the signaling. So conjecture with injection of the IXP2 inhibitor significantly block the protective effect induced by the testosterone, suggests that the protective effect of the androgen is mediated by IXFP2 signaling pathway. The panel A shows 21 days of the testosterone replacement therapy induced 100% testicular descent in the HCG receptor knockout animal. The co-treatment with IXP2 inhibitor prevent testicular descent 50% of the HCG receptor knockout mice placed on testosterone replacement therapy. So these results further suggest that the androgen induce IXP2 expression to sustain the IXP2 signaling. Is thus essential for the completion of the second phase of the testicular descent. To determine whether the androgen induced the genital femoral nerve is involved, the androgen induced the testicular descent in the HCG receptor knockout. That's previously I mentioned that that's been proposed that the androgen might act on the called genital femoral nerve. That somehow the genital femoral nerve somehow derived the second phase of testicular descent. So that's what we performed this experiment. What we did was we transect the genital femoral nerve 5 days before testosterone is replacement therapy began. And then the data shows that regardless there's intact the genital femoral nerve of the genital femoral nerve is transect. The androgen replacement therapy all they can reduce the second phase of testicular descent. So this slide recap the results of this part of study, show that androgen-induced RXP2 expression and activation of the RXP2 signaling pathway are essential for gynecological development during the Inguinoscrotal testis disease. The action of the androgens on the genital femoral nerve, if any, is dispensable. As I showed previously, the microarray analysis showing that XMOC1 is most severely suppressed in the LICG receptor knockout gynecological tissues, and then robustically induced by androgens. This is the microarray data showing that the significant decrease of the XMOC1 mRNA level in the LICG receptor knockout gynecological tissues. And the single injection 24 hours for 24 hours shows that the expression, the XMOC1 is significantly increased. And this real-time RT-PCR also verified these changes. And then the Western blot and also immunohistochemistry demonstrate that the significant decrease of XMOC1 protein in the gynecology of the LICG receptor knockout mice, and then the androgen elevated the XMOC1 protein. The XMOC1, the full name of XMOC1 is a SPARC-related modular calcium binding one. It's a matrix cellular protein that belongs to a SPARC protein family. XMOC1 influences a variety of cellular functions, include growth factor signaling, cell migration, cell adhesion, cellular proliferation, and differentiation. XMOC1 is highly conserved and widely expressed in the embryonic and adult tissue. The protein sequence of XMOC1 is of human share almost more than 96% of models. However, the role of the XMOC1 in regulation of the gynecological development has not been investigated. The first, we are determining whether or not the androgen directly regulate XMOC1 expression in the gynecological tissues. We perform the primary gynecological cells culture in vitro and treated with dihydro testosterone. This result shows that the DHT significantly upregulated the XMOC1 mRNA level. The co-treatment with the androgen receptor antagonist glutamine block the androgen-induced XMOC1 expression in the gynecological cells. In both mRNA and those Western blot also demonstrated this, the DHT also induced the protein expression of XMOC1. The glutamine co-treatment also block the androgen-induced XMOC1 expression. To explore the biological relevance of the XMOC1 in the gynecology, we first test whether or not the XMOC1 affect cell proliferation. So what we did was we isolated gynecological cell and culture in vitro and treated with the XMOC1 is a recombinant amongst XMOC1. And using the MBT ELISA assay shows that the XMOC1 is moderately and significantly upregulated, increase the gynecological cell proliferation. Immunofluorescence study of the proliferation marker AI67 also shows that the XMOC1 treatment increased the cell proliferation. To test whether or not the XMOC1 influence the myogenic differentiation of the cubicular cells. So the isolated cubicular cells was cultured in the myogenic differentiation medium for 3 days. And then the real-time RT-PCR shows that the DHT, XMOC1 treatment for 3 days shows that the XMOC1 induced the PAC7, MYF5, the expression. PAC7 and MYF5 are two well-established primary regulators of the striate muscle commitment and differentiation in several types of the cell, include the muscle cell or non-muscle cells. So these results suggest that the XMOC1 might stimulate the gliomolecular mesenchymal cell to start differentiation into a myogenic precursor to finally to become the master muscle cells. This slide shows time-dependent upregulation of the MYF5 mRNA and protein level by XMOC1 in primary culture of the molecular mesenchymal cells. And this slide shows the time-dependent upregulation of the PAC7 and mRNA and protein expression by XMOC1 in primary culture of the molecular mesenchymal cells. And this slide shows that the androgen also can still upregulate MYF5 PAC7 protein expression once the molecular mesenchymal cell culture in the myogenic differentiation medium. Interestingly, if endogenous of XMOC1 was depleted by, as shown here, we use an XRID to inhibit the XMOC1 expression in the molecular cells. Then the androgen-induced PAC7 or MYF5 protein expression was abolished. So suggests that the effect of androgen is to induce the PAC7 or MYF5 expression is mediated by the XMOC1. So these slides recap the results of this part. The study highlighted the role of the XMOC1 in modulating molecular cell proliferation and differentiation, and we feel XMOC1 as an important downstream molecule of the androgenic action on molecular development during the transinguinal migration of the testes. So I'll acknowledge my collaborators and postdoc fellows for their contribution to the talk I presented today. And this is today's events code. Okay.
Are there any other models for this? The brand only?
Not. The rod has one of the strand of rod has congenital mutation. That also shows that the transinguinal testis disease, and the results also similarly has a molecular developed defect. Particularly is the differentiation of the muscle cell, muscular muscle cell defect. So that resulted in the Inguinoscrotal testis defect.
So in your knockout animal.
What?
In knockout animal?
Yeah, I mentioned that your neck lab, they has generated Yeah, after we published those results, a few years later, they report they generate the molecular specific androgen receptor knockout. So it is very clearly demonstrated that the androgen therapy act on the receptor in the molecular is essential. They also show this the some phenotype is a second phase of the testicular descent defect in our animal model, as well as the molecular selective androgen receptor knockout animal.
So in your LHR receptor knockout animal, we don't expect any spermatogenesis as well as ovulation in female, right?
Yeah, no. Yeah.
They were, I don't know, but you did not do the later on knockout where you could see if you knock out LHR receptor in pregnant.
In our model, mainly it's demonstrated that knockout LH receptor result in androgen deficiency. And that shows that the Inguinoscrotal testicular descent failure is due to androgen deficiency, not directly linked to the LHCG receptor.
But if you give androgen,
We give, and the good thing is our model can be rescued by give back of androgen and then completely recover to the normal phenotype.
but the receptor is still knocked out.
And it's still knockout. So that's
Androgen should not work.
no, that's LH receptor knockout, but androgen receptor is still intact. Only thing just lack of ligand.
Yeah.
So our model, the good thing of our model is we can replenish the ligand to rescue the phenotype. If it's androgen receptor knockout, that cannot be rescued by that, give additional ligand. Yeah. But if you give more clear cut, they do not have any other phenotype except just is Inguinoscrotal male descent of testes.
But even you supplement with androgen in knockout animal, LHR in knockout animal.
Yes.
Gonads will be still small.
Now the testes become bigger, as I showed.
Because during gestation period, the testes are developed, and due to lack of testosterone, LHR.
After you give the testosterone, the testes also become bigger. But still, the male still infertile. Yeah, still infertile. But testes induce testes move from abdominal to the scrotum. That's for sure. So that's we use this model to study the testes descent phenotype there.
The question on the chat there.
No.
Dr. Lee, this is Satya Krishnaswamy. How are you doing?
Good.
Hello.
Hello.
Yeah.
Hi. So that was a great review. Thank you very much. So my question was related to the mutations that you showed early on in the first couple of slides. That's very important for our fellows. Sometimes we have board questions based on that. So I have a patient with Schwarzman diamond syndrome. I don't know if you're familiar with that. It's a SBDS protein mutation in the nucleolus. They have exocrine pancreatic insufficiency, cytopenias, and cystic fibrosis, like a picture with the diabetes. So this patient that I have also had cryptorchidism. I hadn't seen the association, so I was just curious, if there are rare mutations there.
I'm not familiar in these syndromes.
Okay. Okay.
Thank you.
Thank you.
I don't think Dr. Winters is there today.
Yeah, I know. He's in Cambodia.
Probably
Vietnam.
he'll be able to give a better answer.
Yeah.
That's okay. I was just curious because I just saw that, you had all those mutations and I didn't see that particular one. So thank you. It was a very nice talk. Great review.
What's the question here? What? Team check. As the human testosterone or study that you do this. I'm not sure what's adverse effect in mice. We can give short-term injection of the testosterone or the subcutaneous implant of the slow-releasing partial contact testosterone for 21 days. So we observe is they can induce the testis descent from the abdominal location to the scrotum, enlarge the lumbar size of the cremaster muscle sac, also enlarge the testes. However, there's the male still is infertile, but if fertility of those male after testosterone replacement therapy might be due to some other reasons such as sexual behavior and also incomplete spermatogenic process. They are not probably not get the final mature sperm produced by the testes.
Thank you.
Okay, thank you for coming.